Yeah, I know, this is a stupid gimmick, but it's helpful for editing. This time, we're going to be talking about research methods for studying the brain. I found this really helpful. You just heard that whole spiel because you're probably plowing straight through these. But I found them generally to be pretty helpful in just getting a handle on all the different types of brain research and how to do it. So there are two types. We got ways to measure brain activity and more like imaging aspects. So, and these tend to detect when the brain is working rather than like how. The idea here is for a lot of these brain techniques, we are observing the functioning directly. So EEG, so electrocephal... EEG? I practiced before this lecture. Nothing. So electrodes on the scalp pick up electrical signals generated by the brain, while the magnetic... Oh, God. I had all of these, and they just fell out of my brain. So the MEG detects magnetic indications of brain activity, and this image here illustrates kind of how those two aspects work together. So the electric current generates a magnetic field. And so the MEG detects this technically indirect effect, but it's pretty closely linked. I mean, one made the other. So here it's detecting when the brain is working. So for the EEG, it's a surface recording of electrical activity in the brain cells. So there's lots of different ways to place your electrodes. Between 10 and 12, these are connected to the EEG machine. Oh, that's bad. I'm usually at least good at plowing through it. But what it does is it amplifies the brain activity signal so that it can be recorded. Otherwise, it's like these tiny little blips that so you can't just like hold up a magnet and be like, oh look, it's moving. No, you have to really magnify the signal to be able to detect it. And here's a cool little diagram that looks at the analysis schematic for each different burst. So here's the basic EEG signal. The most kind of the interesting piece is that burst, right? That high spike, as well as kind of the timing between them. So there are different techniques for how to extract this information. The bursts tend to be binned into several groups based on duration, and as we can see in this illustration, the average shapes for each group are kind of calculated, and there's a lot of data management that goes in that you don't care about, but I think is really neat. And they examining these shapes they're analyzed for cool things like skew, kurtosis as a function of duration, and alright, yeah, I could go on and on. But the idea here is that it's not just getting the direct measure. There's lots of processing involved, and as you can also see, there are lots of different ways to collect this data, going from infants where they just wear a hat to kind of a more specific node placement that goes directly on the scalp, as well as for kiddos where it's a hat but it's not as babied. So here's the EEG. Next, we got the MEG. I'm not even going to try. So this is a functional neuroimaging technique for mapping brain activity by recording the magnetic fields, which are produced by the electrical currents that occur naturally in the brain. So you have to use really, really, really sensitive magnetometers. And I've illustrated a couple of different methods here, ranging from just generic adult to adolescent to here's a kind of a close-up of a baby one. Here's it with a baby in it. And this one took me a little while to figure out exactly what was happening because I saw this picture and I was like, what? That is a pregnant lady. They are measuring her baby. They're measuring the baby's magnetic pulses, not like the mom's just like stomach contents. Seriously, I stared at this figure for this little one right here for a little too long, just to be like, because it needed some context. Cool. Okay, so we had 2 that looked at when parts of the brain are working. Now I'm going to talk about detecting what parts of the brain are working. So we've got computerized tomography, so these are CT scans. They can examine really, really small structures. We've got positron emission tomography, where we map brain activity based on blood flow. That's a PET scan. And we've also got the fMRI, which maps brain activity based on magnetic impulses generated by oxygen in blood. So these all kind of, and jumping back, so the CT scan are computer processed x-rays. So they can give you a decent snapshot of like what stuff is in there without having to cut anybody open. And so x-rays, brain flow, blood flow, and then magnetic impulses generated by blood flow. All these kind of give you a sense of what structure. And I've got illustrations for all of them because that's fairly helpful to see, because usually you'll read about, just a CT scan or a PET scan or an fMRI scan, and your brain just sees an acronym and it's like, brain stuff. So for a CT scan, it used to be called a CAT scan, CAT for computerized axial tomography scan. They shortened it because I don't know, but that's one reason why you'll hear some folks say CAT scan, others say CT scan. They're the same thing. These are computer processed x-rays, and they create cross-sectional, tomographic images, essentially virtual slices. And it allows you to see inside without cutting. Here, we've got a picture of a 14-year-old lion named Tomo, who they at the Columbus Zoo in Ohio. They were looking to see how effective, if he had a fungal infection. I mean, they had a good sense that Tomo had a fungal infection, but they wanted to know if it had spread beyond his gums, and whether they needed to do more than just give a 2-month course of antifungal medications, just to get a sense of looking in there to help with diagnosis. Okay, so I've also got some more coming up to give you a better additional sense of what's going on. So here's some more images of CT scans. You don't need to just apply them to living people. They're great for mummies and rhinos. I had some fun looking at non-human cases, because it's fun to me. Now here, I don't know how well this animation will capture, are computer composites generated from CT scan data. So we have individual slices here and here and here, but you can kind of rebuild them together to create a better illustration, to recreate what you're seeing instead of just looking at weird snapshots. Not weird, but I find it helpful at least, and then there's some cool things you can do data visualization wise, and animate it like this GIF here to kind of see the structure in a way that is a little more intuitive, because while this cross section of a human, if you hadn't told me this was a human being, I don't know it looks more like a Rorschach test to me. A couple of these. I mean, this one's got some spine bone stuff, but in certain slices, it's really hard to see. And so I find it really helpful when you do these extra kind of animations. Now, a PET scan, this is the one with a lot of the cool bright lights. It's positron emission tomography. So what they do is they use a radioactive drug, it's called a tracer, to show activity. And here we've got a picture of a normal brain up here. You can see it here. as well as kind of kidneys and whatnot, as well, and radioactive urine down here. In addition, we've got, okay, well, here's what my notes say, and we're gonna read those and not just have me like point around. So the normal brain and kidneys are labeled somewhere, apparently not. Labels. I think there might be an animation. Whatever. Enjoy my struggle. So the normal brain and kidney are labels, and the radioactive urine from the breakdown of the FDG, so the tracer, is seen in the bladder. Now we've got a big metastasized tumor mass from colon. Oh, that's the tumor. That's colon cancer. That you can also kind of see in the liver. Yeah, so this was a tumor investigation. So here, based on the accumulation of the tracer in the heart, bladder, kidney, and brain, liver metastasizes to lots of parts of the image. So here's the picture one. Essentially, here's the one that gives you the cool, bright colors and kind of illustrates where there's more blood flow, relatively speaking. So less blue, less blood. It goes from blue to red in the way that looks pretty obvious here. Cool. So here's an illustration. This is a little out of date now, but I thought that the Zika illustration was fairly useful. So here, and also you'll notice that a lot of these measures can be combined. So here in this Zika image, this PT/CT scan of a mouse looks at infection of the Zika virus. Well, here's the one where no control. So here's the control mouse. Here's the Zika mouse. And you can see that essentially it heats up. Then there's lots of neuroinflammation by a little on day 6, ton on day 10 to kind of illustrate this. Also, a lot of the machines that, oh look, how convenient. I forgot that I have an animation. So, yeah. A lot of these machines will kind of combine measures into like this big thing because you can do multiple measures at once, which is kind of nice and convenient. Yeah, but the big one, the one that we do tons of stuff with, the one I've worked the most with data, is functional magnetic resistance imaging, fMRI. So this measures brain activity by detecting changes associated with blood flow. So this exploits the link between cerebral blood flow and neural activation. These are associated strongly. They're not perfect. But when an area of brain is in use, blood tends to flow to that region. And as you can, in these diagrams, similar to how the PET scan coloring works, darker or redder colors indicate increased blood flow. Blues indicate reduced blood flow, and that's kind of the idea. And you can see where the blood flow is, and it kind of identifies the structures that are likely being used, or at least are drawing in more blood, to examine responses to faces or other stimuli and just seeing how they differ. So here's a graphical summary of an fMRI data for sensation seeking. So sometimes like we'll do a study where we just like we compare groups. Now I don't always encourage this with a lot of measures that you essentially have to like slice because it has the same problems that the Myers-Briggs does, but with grouping and losing the people in the middle. However, it's still pretty useful. To give you a sense of if there are differences. So here we can see that for those that are sensation seeking, so here's the high sensation seeking, comparing wins versus not winning. There's brighter, deeper differences, and more stuff is kind of activated versus low sensation seeking. There's less orange, relatively speaking, and just less stuff's lit up. And So they show distinctive patterns in brain activity during reward processing. That's the basic idea. These tend to make really great figures in general, and they give you a nice sense of what's going on. And they're usually, the fMRI data is usually overlaid with either like a standard brain image or from CT scans. So here are some pictures of, I've been really interested in seeing like how different ages go into these, the fMRI. And so here we got a super baby with soundproof headphones as much as possible. And here's a less of a baby. This, you could argue, is an adult. Anyway, yeah. So same kind of setup, a little smaller and more swaddled for the kid. But I find it helpful to look at all the different images and see it. Now, there are a lot of difficulties with imaging techniques because, like blood oxidation may inhibit activity that's inhibitory in nature, so not just like excitatory. There's a great footnote detailing this on page 266. And so, like, blood flow can also increase when your brain is saying, like, slow down. Like, so it's not that blood flow is always positively associated with, like, more activation. All we know is that it's changing. So, in some part, like, all parts of the brain are kind of always active to some extent, and so blood oxidation level dependent image, like, so there's this thing called bold signals, and these look at how activation differs from neutral or a control group across brain imaging, and they find that even those light up. So it's really helpful to have like baselines because they don't always have the baselines. And just because like an area of a brain is active in response to a stimuli doesn't mean it's the same for everybody. It's like not, it doesn't have to be the same psychological process that occurs each time in the area. So there's a great example in the textbook of emotion. And we're making some pretty steep assumptions, like that these line up. Now, I am not a neuroscientist. They do check these assumptions, but from the statistical side, I have some insight. And some of the biggest difficulties are like, well, first of all, most researchers only focus on a really small area. So same as how like in personality psychology, we look at various paradigms and kind of focus on specific parts. The downside of that is you can miss important areas outside of your narrow scope. So one big theory can be helpful when it comes to data collection. If only we had that. So there are some downsides there. Now, the technology is really difficult to use. It requires specialized training, so you can't just like send in, well, you shouldn't at least send in like untrained undergrads to run research subjects. It's expensive as well. I'm blanking on the number right now, but it's like multiple $100s per hour for data collection. There's also this weird quirk, so it's called the neural context effect, where activity in one area may depend on what's going on in another more other areas. So that's another aspect that you may miss out on the interaction between pieces if you only focus on one tiny little area. So jumping back to those tech difficulties, so even the technology is difficult to use, but also the statistics used to summarize are these like really complex data the statistics are complex and can be inaccurate and may lead to misleading or exaggerated results. And the statistical modeling is really in its infancy. There's lots of ways that we can improve. I actually recently reviewed a paper this summer that kind of applied some of the statistical techniques that we use in modeling genetics to look at kind of SNPs, like so small chunks of DNA, and looking at how those map onto voxels. So the various big slices of DNA, because of a brain. So when we're doing these fMRI studies, we're taking chunks. We're not like, the resolution's not super tight. It looks like it's super tight, but we're measuring like little cubes. And so those statistical techniques are similar, which is kind of cool. And I wrote really nice things about that paper. So hopefully it will come out. I think it was a good, it applied the two methods and linked them and looked at brain by gene interactions, which are really, really cool. And we'll talk a little about next time, because next time we're going to talk about the brain some more. I know, well, actually, I've talked all about the methods, and you'll like the method section. This is what this lady does. She likes her stats, and so we indulge her. But yeah, so next time we're going to talk about the brain in specific pieces. And yeah, I'm going to cut it here because I've been talking for... I try and record some of these lectures in chunks. But yeah, I've been talking for quite a while now and I'm a little pooped. So I'll see y'all later. Bye!